Welcome to today's Explainer. We're jumping right into a really fascinating intersection of cellular biology, genetics, and clinical eye care. We're going to trace a journey that starts at the absolute foundation of human energy production and ends with a profound clinical condition that can alter a patient's life literally in an instant. If you've ever been curious about how a single, tiny mutation in our microscopic cellular power plants can systematically dismantle someone's vision, well, let's dive right in. Here's our roadmap. We'll start with the mitochondrial power plant, then move to expressions of dysfunction, early loss energy crisis, and late loss toxic stress. After that, we'll put a spotlight on LHON and wrap up with LHON clinical characteristics.
Section 1. The mitochondrial power plant. Let's start right at the source. So I want you to imagine the mitochondria as a microscopic power plant. Inside, there's this machinery called the electron transport chain, which drives protons out to synthesize ATP. Remember, ATP is basically the energy currency of our cells. This entire process happens by passing electrons through 6 specific membrane elements. We've got 3 transmembrane proteins, complex I, complex 3, and complex 4, along with 2 surface proteins, Cytochrome C and coenzyme Q. As electrons float on this chain, it pumps protons, or H+ ions, out. Those protons then drive ATP synthetase, literally manufacturing the energy required to keep our highest demand tissues, like our muscles and our retinas, up and running. But here are the stakes. What happens when this machinery springs a leak? Well, instead of producing energy, it causes oxidative stress and widespread cellular damage. You see, the electron transport chain isn't totally flawless. Electrons can actually escape or leak out at the protein complexes. This happens most often at complex I, then complex 3, and finally complex 4. And when these runaway electrons bind with ambient oxygen, they create superoxide. That's a highly potent free radical. So a leak doesn't just mean you're losing power because you're failing to synthesize ATP, it also means you're directly pumping toxic oxidative damage into the surrounding cellular environment. Section 2. Expressions of dysfunction. Let's see how this cellular biology translates into actual physical disease. We can break this down into a really helpful rule of 3. There are 3 primary ways a breakdown in our cellular power plant expresses itself. First, you have an underlying anomaly right in the mitochondrial DNA. Second, there's an energy deficiency coupled with myopathy, and this is specifically tied to early complex loss, so breakages at complex I or 3. And the third expression is oxidative stress causing neuropathy, which stems from later complex loss, primarily happening at complex 4 or Cytochrome C. Section 3. Early loss, energy crisis. Let's focus on that early break first. This is the true aha moment of our explainer. Notice the critical difference here. An early break in the chain causes a massive energy crisis for high-energy tissues, while a late break creates toxic pollution that destroys delicate nerves. When dysfunction hits early, at complex 1 or 3, the sheer loss of ATP production completely outweighs the oxidative stress. Tissues that are incredibly hungry for ATP, like our muscles and the retinal pigment epithelium, or RPE, begin to fail, leading to myopathies and RPE disorders. But if the breakdown is late in the chain, say at complex 4, the resulting oxidative toxic stress far exceeds the actual ATP loss. This toxic environment drives up apoptosis, which is programmed cell death, and aggressively targets neural tissue, causing neuropathy. To really understand this early loss power outage, let's walk through the escalating myopathies it causes. It starts with CPEO, or chronic progressive external ophthalmoplegia. This develops over years. Sometimes you literally only notice it by looking back at old school photos. It presents with ptosis, though interestingly with the retained lid crease, which helps differentiate it from myasthenia gravis. It also brings ophthalmoplegia and a salt and pepper pigmentary retinopathy. If you took a muscle biopsy, you'd find ragged red fibers where the diseased mitochondria clump together. Stepping up in severity is Kearns-Sayre syndrome. This has all the visual signs of CPEO, but adds severe systemic issues like heart block, hearing loss, and endocrine disorders. And finally, MELAS. This is devastating, adding catastrophic systemic conditions like lactic acidosis and childhood strokes, often before a patient even turns 15. Section 4. Late loss, toxic stress. Now let's pivot to the fallout from those late chain leaks. When that late-stage toxic oxidative stress bombards the optic disc, it results in advanced neural apoptosis and optic atrophy. The normal optic disc progressively dies off, usually becoming visibly pale by the time a patient is anywhere from their teens to their 40s. Because this neural tissue is just being hammered by free radicals, we aren't just looking at structural weakness. We are looking at aggressive, localized cell death. Section 5. Spotlight on LHON. Now we get to apply all of this biology to a very specific real-world clinical condition. We are talking about Leber's hereditary optic neuropathy, or LHON. This is absolutely crucial to know, as it is the most common inherited mitochondrial disorder we see. Now, because mitochondria are passed down exclusively from the mother, a positive family history on the maternal side is a massive red flag. On a genetic level, LHON is driven by specific point mutations in the mitochondrial DNA. To get granular with the numbers, the main culprits are point mutations at positions 11778, 3460, 15257, and 1444. These precise genetic errors are exactly what set the stage for that late complex loss and the toxic cascade we were just discussing. Section 6. LHON Clinical Characteristics. So, what does this actually look like when the patient is sitting in your chair? Well, first, there's a staggering demographic reality to LHON, a 9-to-1 male-to-female ratio. 90% of those affected are male. Think about that for a second. Even though it is completely maternally inherited, it overwhelmingly strikes young men. This heavily skewed demographic is honestly one of the most defining and frankly baffling characteristics of the entire disease. In the clinical presentation is devastating. This profound vision loss strikes young men right in the prime of their lives, typically between 20 and 40 years old. They experience a progressive vision loss that can drop visual acuity from around 624 all the way down to absolutely no light perception. Now, it often mimics the symptoms of optic neuritis, but with one absolute key distinction. LHON is completely painless. It usually starts unilaterally in just one eye, but it inexorably progresses until it becomes bilateral, blinding both eyes. Research has shown us that LHON specifically targets the retinal ganglion cells. It reduces their ability to produce enough energy to survive the oxidative barrage. Current management focuses heavily on understanding this pathophysiology, trying to prevent onset in known carriers, and exploring free radical scavengers and gene therapies once the vision loss starts. But here is the wildest part. These mitochondrial DNA mutations are entirely silent. A carrier can go decades living a completely normal life without any visual impairment whatsoever. Which leaves us with one of the most lingering genetic mysteries in neuro-ophthalmology. If the mutation just sits silently in the cellular power plant for 25 years, what exactly pulls the trigger? We know the genetics, we understand the cellular mechanisms, and we clearly see the devastating clinical outcomes. But the exact biological or environmental spark that suddenly activates this silent mutation in a perfectly healthy young man, we still don't know for sure. It's a huge puzzle, and it just goes to show how much we still have to discover. So keep asking questions, keep digging into the science, and thanks for joining us for this explainer.